Okay, so for those of you who don't know me, I'm Dr. Sarah Dyke. I'm one of the clinical geneticists at the IWK, and it is my great pleasure to introduce Dr. Sean Myles to our gathering here today. I became aware of Sean when I was listening to the radio on my way into work, and he was, well, it was on the weekend, taking the kids somewhere, who knows, but he was on Quirks and Quarks, and I was really interested in what he was talking about. It was about human genomics and gene discovery. And then to my amazement, it said that he was from the Agricultural College in Nova Scotia, so I felt I had to, find out a little bit more about Sean, and thankfully he was willing to come and talk to us today about some of his human genetics work. So he's originally from Fredericton, New Brunswick, where he received his BA at St. Thomas University. He completed a master's in human biology at Oxford University and a PhD in human evolutionary genetics at the Max Planck Institute for Evolutionary Anthropology in Germany. He turned to plants during his postdoc at Cornell, where he worked in Ed Buckler's lab on grape genomics. He also spent time as a postdoc in Carlos Bustamante's lab at Stanford before starting his current position at the Faculty of Agriculture at Dalhousie University as the Canada Research Chair in Agricultural Genetic Diversity in the summer of 2011. His research focuses on elucidating the domestication and breeding histories of apples and grapes and figuring out how to use genomics to more efficiently breed and prune fruit that requires less chemical input to grow. So those of us who enjoy a glass of wine will be eagerly watching his results. And improvements, will be experiencing his improvements, no doubt. So to explain how a CRC chair in agricultural diversity can add to our knowledge of human genetics, please welcome Dr. Sean Myles.
Thanks for the nice introduction. Yeah, I was fortunate enough to be on Quirks and Quarks. As a young scientist, you're growing up, you always think, like, when I really make it big, I'll be on Quirks and Quarks. And I feel like, like exhausted. So early in my career, I was fortunate enough to be on Quirks and Quarks and already, achieve the great status of Quirk and Quarkdom that I thought. But no, it's been great actually. I love the program, so. It's been good for me to get the name out of what we're up to. So I do work on apple and grape genetics, but today I'm going to talk a little bit about what I was up to mostly during my postdoc and after my PhD and so on. And I picked a title that I thought would attract attention and get people to come to a talk, and I think maybe it worked because it seems pretty full. I'm just going to talk about a project that is hopefully relatively entertaining and will be some fun. So. In general, what a lot of people are interested in the genomics world, of course, is establishing relationships between genotypes and phenotypes. And we do that through a method called genetic mapping. And many of you are familiar with genetic mapping. There are two types of data that you need. You need genotypes and you need phenotypes. We're all familiar with lots of different phenotypes, especially the medical genomics folks, trying to get BMI, or you're looking at ovarian cancer, or you're looking at type 2 diabetes, or whatever it might be, or height in this case. But the genotype data really, it used to be a big bottleneck in genomics, and more is becoming very easy to collect. So I'm going to talk a little bit about the genotype data, and then we'll talk about the phenotype that we're going to talk about today. So the genotype data I'm going to talk about are in the form of single nucleotide polymorphisms, which are, I think many of you are familiar with, the most common form of genetic variation in the human genome. And the basic idea, your genome is made up of A, C, C's, and G's, and as you cruise through, you look for positions that differ by one single nucleotide in the genome. It looks something like this. So at this single position, one individual carries a T, and the other individuals have C. This is called a SNP. So that's what I'm going to be referring to, SNPs. And what we want to do to collect genotype data, we want to get a lot of it. We want to get a lot of these SNPs across the genome. So in theory, what we want to do is take a bunch of individuals and go through their genomes and identify all the positions that differ by single nucleotides. And that used to be really challenging. Now it's really quite simple. You generally, if you're doing the types of studies that I'm going to talk about, you buy a commercially available genotyping microarray and you put DNA on it and you get a result back. Between 500,000 and 1 million of these SNPs will be genotyped with one of these arrays. And increasingly we're seeing now in the literature that next generation DNA sequencing is probably going to blow arrays out the door, and then everybody's just going to do next gen sequencing with some imputation, and then you're going to have your genome-wide SNP data. So really what we want is to saturate the genome with these SNPs, genotype a whole bunch of them across the genome for a whole bunch of individuals, and then you collect a phenotype, and then you look for an association between the genotype and the phenotype. This is very misleading in terms of how many SNPs we've just found in this little tiny region. In general, in humans, if you take a group of humans like us in the room and we take a look at our genomes, we only find one of these SNPs about every 1,000 base pairs. So these are the types of data that we want to collect, and it's becoming cheaper and cheaper to do this. But the real question is, what kind of phenotype are we going to look at? And so this is the fun part. We're going to choose a really cool phenotype to look at, and it's blonde hair in Melanesians. So a lot of people think that blonde hair is exclusive to Europeans. But it turns out that there are people in the Pacific, on the coastal Papua New Guinea, out in Bougainville, Solomon Islands, Fiji, Vanuatu, even Australian Aborigines who have blonde hair. They're some of the darkest skinned people on Earth, yet they have this striking blonde hair phenotype. And I've had a very fortunate scientific career. I did my PhD at this Max Planck Institute, and one of the first days I was there, the nervous young grad student gets called into the supervisor's office, and he calls me up and says, "I want you to come to my office." And I think, oh, boys, I think I've done something wrong. And he sits me down and he says, "So, do you want to go to the Solomon Islands?" And I said, "Yeah, that'd be great." He said, "Yeah, well, we've got this project going, I'd like to go to the Solomon Islands, we'll go and collect DNA samples, and we'll do a population genetic history study of the Solomon Islands in the Pacific." I thought, wow, this is really great. And the first thing I did is ran back to my office and Google Maps Solomon Islands. I had no clue where I was. I mean, this could be like in the Indian Ocean, or this could be down in the Antarctica or something. But, so Googled it, found out where it was. So we went there many, many years ago, and when we were there during that time, the goal was not to map the blonde hair phenotype. We were doing just a general population genetic study, looking at mitochondrial DNA and Y chromosome variation in the Solomon Islands and in the Pacific. But while we were there, we noticed this striking blonde hair in some individuals, and I thought to myself, man, we got to map that. Somebody has got to find the gene for this. This would be just a really cool thing. And when you ask around, you ask people what causes the blonde hair, they say, oh, it's because some kids, they go out into the sun, and the sun causes their hair to go blonde. And I'm thinking, no way, this is definitely genetic, And then other people are saying, well, it's probably introduced from Europeans, right? Early European explorers came by, dropped off a couple genes at the door, right? And this is what ended up showing up. Certainly wasn't due to our expedition. That would be too recent. So this striking blonde hair phenotype, anyway, is clearly very attractive. It's a visible human phenotype, and it would be really interesting to take a look at evolutionarily. So we decided to go and do that. So I applied for a grant. I got some money to go back. We went back about 5 years later. This is where the Solomon Islands are, by the way. So here's New Guinea. And so there's a group of islands off the coast, and it's a beautiful place. And I thought, it's probably minus 18 degrees outside, so I give you an idea of what it's like cruising around for 6 weeks in the Solomon Islands in a little canoe and going off to these isolated little islands. Everything moves, and when you're doing this type of field work, very, very slowly. You can't just row up to an island and expect people to line up like a clinical trial and be extracting samples like crazy, super fast, and everybody filling out forms, because most people can't write. A lot of people don't know their birthday. You ask them how old they are, they say 50. And you ask somebody else, they say 60. You should see our age distribution. There's no curve to it. It's like people are either 40, they're 50, or they're 60, because they're kind of just estimating by decade, how old they are. Right? So dealing with indigenous populations and going out and sampling like this is very, very difficult. The ethical issues surrounding going out and sampling, there's a massive number of hurdles and that kind of stuff. So going through RIVs took a lot of the IRB approval and stuff. That took a lot of time, but we eventually made it out there. And we had a lot of fun. We meet a lot of really nice people who help us out. But again, you got to be very, very patient and not expect things to happen too quickly on Solomon time, is what they call it. So we have a boat that we cruise around to the different islands, and I went with a colleague of mine, Nick Timpson from England, and we ate a lot of fish. And so you cruise around from island to island, and you meet with the communities, and generally they have a chief, and so you explain to the chief. Luckily they speak English, so they have a pidgin English, so you learn enough pidgin English that you can explain the project and that kind of thing. Some of the outlying islands there weren't very many English speakers, so you have a translator with you and that kind of thing. But generally it takes a very long time, but eventually you get people lining up to participate. If they wish to, they spit into a tube. Here you can see the people sitting down spitting, providing saliva so that we can get their DNA, and then we measure several traits. We take down some information about them. And we measured their hair pigmentation using a spectrophotometer. We also, at the same time, took a whole bunch of other measurements. We took height, weight, hip circumference, waist circumference, head circumference. We took skin pigmentation also underneath the arms. We did, and blood pressure. So we thought, you know, while you're there, do everything that you can possibly do out in the field easily. Places that are easy to sample, hospitals and schools and things like that, where people are used to lining up. Because on the outlying islands, it's a lot more difficult to get people even to come together out from down from the fields. You know, you're waiting 3-4 hours, you know, just hanging out. So, so yeah, the phenotype is definitely very striking. You can tell this blonde, wavy hair that you see, particularly prevalent in children, and it comes in all sorts of different different shades and things. So it's a really fascinating phenotype in general, hair. When you think about it in terms of, you know, of all the different species on Earth, humans have this incredible variation in hair, not only texture but also color. So, and so you can see as people get older, we notice their hair does get darker. So the phenotyping in the kids was really unambiguous. You could really distinguish blonde from non-blonde quite easily. And here's some more sort of like, you can see variation in hair texture as well. You know, you get this straight long hair, and also very curly hair. So what we're interested in doing is mapping this trait. We want to find the gene for this trait so that we can say something about the history of blonde hair in Melanesia. Excuse me. So how do we do that? Well, the easiest way, we do what's called a genome-wide association study. So we have these commercial genotyping microarrays where you can collect 500 to 1 million SNPs. And so we pick, the way that we did it, we picked, how many do we have? 42 blonde individuals and 43 dark-haired controls. Okay, so it's a case control study. You genotype the blonde individuals, you genotype the dark-haired individuals, and then for every single SNP in the genome, you've got one of 3 possible genotypes, right? You're either homozygous for the reference allele, you're heterozygous, or you're homozygous for the non-reference allele. And so you're either 0, 1, or 2. And so you code every individual by 0, 1, or 2, all the blonde individuals, and then you look at 0, 1, or 2 for all the dark-haired individuals, and you run a chi-square test all the way across the genome, and you just plot the p-values for every single SNP that you've genotyped. And here we genotyped 600,000 SNPs with the Illumina array. And each of these dots represents a SNP in the genome, and here you have your chromosomes, and this is called a Manhattan plot. You have the minus log 10 of the p-value here on the y-axis. So this is the association of each of these SNPs with hair color, okay? With the case control status, the blonde or non-blonde. And this dotted line here represents a kind of an arbitrarily, not totally arbitrarily, it's Bonferroni corrected p-value threshold. So anything above, any dots above this line suggests that it's highly significant, and it's a significant hit. So as you can see, there's one region of the genome that pops out like crazy, and this never happens. Because these are called Manhattan plots. They're supposed to look like Manhattan, right? They're supposed to be logical. I call this the Dubai plot, because there's one big hit right in the middle. So, and, you know, as a researcher, you go and you think, you know, you hope for a large effect allele in the genome when you're running these genome-wide association studies. You're hoping, yeah, I hope there's like one gene that causes type 2 diabetes, and we know that's not true. But when you're looking at blonde hair and you have no idea what the mode of inheritance is, you know, it may be one large effect allele, so it really definitely looks that way. It looks like there's one region in the genome that's responsible for being blonde or non-blonde. The odds ratio is ridiculous, right? Your p-value is 10 to the minus 19. In fact, we didn't need to genotype that many individuals. We could have saved ourselves tens of thousands of dollars if we could have done it with 6. You know, 6 cases, 6 controls, and we would have been able to map it. So in the end, though, you see that there are many, many, many dots here that exceed the threshold, right? And so on these genotyping microarrays, you're only genotyping the SNPs that can be assayed by the array. And those SNPs that are assayed by the array have been chosen by Illumina from databases, and generally those databases are populated with individuals of European origin, or mostly of European origin. So the chances that the SNP that causes blonde hair in Melanesians is on this array is basically 0, okay? Because first of all, we don't know which SNP it is, or even if it is a SNP. So all of these SNPs that you find within this region are in linkage disequilibrium. That nasty term we learned at the beginning of our genetics careers. It just means that they're all correlated with each other. And the region of the genome that we're able to nail it down to here is about 400,000 bases wide. Okay, so you get this 400,000 400,000 base pair region flanked by 2 recombination hotspots, and you're thinking, okay, 400,000 bases. Where within this 400,000 is the single nucleotide causing blonde hair and Melanesians? Right? So we've narrowed it down quite a bit from 3.5 billion down to 400,000. That's pretty good. Now we want to do some fine mapping, if you will. So what's the first question you ask when you're thinking, okay, what's the next step? Right? Well, let's take a look within this 400,000 base pair region and see if there's any genes in it. Is there anything? The lowest hanging fruit is going to be something that's protein coding. Is there anything protein coding inside this region? Most of the human genome isn't protein coding. So every once in a while you do see a gene. Turns out there's only one gene in that entire 400,000 base pair region. And oh geez, there's only one gene. Let's go look what it does. And PubMed TYRP1. What does it do? It's a tyrosinase-related protein, and it's involved in mammalian pigmentation. Known mutations in this gene cause albinism in humans. They cause lighter coat color in the mouse and in the horse and in hamsters and rabbits and all sorts of things. So you think, okay, this is a pretty darn good candidate gene so what's the next step? Well, we should sequence this gene. So let's take 12 blonde individuals, and we'll take 12 dark-haired individuals, and we'll just sequence the exons of this gene, the protein coding sequence. And let's see if we see any changes that the blonde individuals have that the dark-haired individuals don't. And everybody's going to be jealous because science never works this way. But so there's one position in the entire amino acid sequence that differentiates the blondes from the non-blondes. So here's the amino acid sequence around exon 8 of this gene. And I've put in the multiple sequence alignment here across species just to show you that this is a relatively well-conserved region of the protein. So it's been conserved over a very long evolutionary time. And this one position here, you can see that all 12 blonde individuals at this position, when we sequence this in 12 blonde individuals, they're all homozygous TT at this position, while the 13 controls are either heterozygous CT or homozygous CC. So that suggests that the T allele, that you need to be homozygous. So it suggests that the T allele, you might be recessive, blonde hair may be recessive, right? And that you need the two T alleles in order to be blonde. And that T allele there, it changes in the amino acid sequence, so it's a non-synonymous SNP. It changes the amino acid sequence of the protein. And it changes this arginine to a cysteine at position 93 in the protein. That position 93 happens to be within a known functional domain. And that arginine to cysteine, you can see arginine has been conserved all the way back to chicken for millions of years of evolution. And that suggests to us that chances are, if you change it, it's probably going to have an effect. It's probably going to have some kind of function. So everything's pointing towards this being, like we nailed it down to a single nucleotide. Right? And then we say, are there any other mutations nearby that have known effects? What's funny is that just downstream here at position 38 in the amino acid sequence, when you change the mouse from an arginine to a cysteine, the same amino acid change, you get like a light blonde colored mouse. Right? So we didn't even have to make a mouse model to prove function or provide evidence of function. We already had something, a nearby amino acid change of the same nature causing both. So we're thinking, we're not proving that this is the nucleotide change that causes blonde hair and Melanesians, but if you're ever going to provide evidence for something, we've got a pretty strong evidence that this is the case. So I know that that's not usually how genome-wide association studies work, and people who are familiar with them, you open up Nature Genetics these days, and it's just like these 400 authored GWAS studies, one after the other, and you look in those, and they're Manhattan plots, like things barely creep over a significance threshold, and they're not really sure which nucleotide is causal, but they've got a couple of flanking markers, and so this was a real exception in terms of. But one of the reasons it is an exception is because we went somewhere that nobody else is going. We did a genome-wide association study on a population that people aren't looking at regularly, and looking at a trait that people haven't really looked at. So in the end, we took our entire cohort of 1,200 individuals that we had both DNA and phenotype data for, and we genotyped them for that particular SNP that we discovered. And we find that individuals who are TT, here is normalized hair color corrected for sex and age on the y-axis. And you can see individuals who have the TT genotype generally have blonder hair or lighter hair color. There may be a slightly codominant effect here because the heterozygotes are lower than the homozygotes here. But in general, it looks like a recessive mode of inheritance is the best model that fits it. And we explain almost 50% of the variance just with this single nucleotide. So 50% of the variance in hair color in this cohort is explained by just genotyping one SNP. That's very unusual. I mean, you don't explain 50% of the variance in BMI, with whole genome sequence data, as far as I know. So this really does suggest it's a very large effect. The frequency of the allele in the Solomon Islands is 26%. So that's the frequency of the allele. And when we go and we genotype that particular position of the genome around the world and try to find out, did that allele come from elsewhere, we don't find it anywhere else in the world except in Melanesia. So this suggests that no, this allele was not brought in from European travelers. This is an indigenous substitution that occurred in the ancestors of Melanesians and has risen to a frequency of 26%. A lot of people will ask, well, did it rise in frequency to 26% because it was under selection? Was there any advantage to having this particular allele? Were blonde-haired individuals favored? Did they leave, on average, more offspring over many generations? And we look at the data as best we can with the data that we have to look for signatures of selection at this locus, but it turns out that that's very difficult to do when the trait is recessive. Because what happens is all of our models for detecting selection are the allele arises in the population and it's immediately selected for, right? But with a recessive trait, the allele arises and it's in a heterozygote, right? And that heterozygote, and then most of the time, for the first many, many generations, it's found almost always in heterozygous form. That means that the phenotype doesn't even show up. So there can't be any selection on it, on the phenotype. Right? It's not until it shows up in homozygous form that you get selection. And while it's drifting around there in the population in mostly heterozygous format, it's recombining onto different haplotypes, and you're losing the power to detect selection, because what we expect with selection is that an allele arises, there's one big long haplotype that just goes and sweeps through the population, and we don't see that. So we don't see any signature of selection, but it's very difficult to test with a recessive allele. So we don't answer the question about whether it was advantageous to have blonde hair in Melanesia, but we do answer the question of that it did arise in Melanesia. It's an indigenous Melanesian allele. How widespread it is, we didn't have any Australian Aborigines to genotype, so we're not sure if the blonde hair that arises that you see in Australian Aborigines is actually the same as the one that you see over in Melanesia, in Papua New Guinea and in the Solomon Islands. So that's basically the story about the Melanesian blonde hair. But how does this relate back to the title of the talk, which is about current medical genomics doing more harm than good? I think everybody's familiar with the fact that we're moving into an age of personalized genomics, and that we just had a baby actually a couple weeks ago, and it's probably I wouldn't be surprised that she'll have her genome sequence available to her physician before she's 6 years old, say. That wouldn't surprise me at all. By the time she's going to school, she'll probably have the physician should have access to her genome. And we're using these predictors of sort of did your father have any problems with what did your father have any diseases? Does your mother have any diseases? What about your mother's father? What about your mother? Eventually we're just going to look at their genome and try to get these predictions. And there are a lot of people after this kind of information. But in order to get that information, we know what we need. We need to do these genome-wide association studies. Right? And so people are going to be looking to make predictions about what traits an individual is going to have and what kind of preventative measures you can take and recommend as a physician to these individuals, what kind of things you can recommend to prevent these diseases or to take action in terms of, we know, I don't know a whole lot of examples of these personalized medicine things now, but the ovarian and breast cancer mutations that you can screen for right now, and then you can suggest that people are more diligent about going for self-examinations because they are at higher risk, right? And in fact, this age of personal genomics is already happening. I thought I'd show you my results from 23andMe, but I don't really take a whole lot, I don't take them too seriously because I know how their algorithms work, but I thought I'd show you my elevated risk for a number of diseases according to the genotype data that I get from 23andMe. So you spit into a tube and you send it to this company, 23andMe, and they'll go and genotype you with one of these arrays, And then they'll give you, they have 4 stars of confidence in this, which I'm sure is a really great model. And they tell me what my risk is. It's actually pretty good. If you go in, they do cover their bases when you look at the fine print in terms of what all their models are additive, right? So they just say, there are 10 risk alleles that people have discovered, and you've got 6, so you're like 60%, So and if you had all 10 risk alleles, you'd have X amount more. So they're really quite simple, and they do say that. But this is already happening, right? I'm already able to go online and check as new results become available through genome-wide association studies. I can go online and I can check, hey, did my risk of atrial fibrillation, did it go up last week, or am I still at the same risk? Because I look like I have an elevated risk. I go to the gym way more now because of this. No, I don't really take any action, but you can see how the way that it's moving, right? And it's genome sequences are going to be routinely, everyone's genome is going to get sequenced, right? Now I say that, everyone's genome. Do you think the guy in the Solomon Islands is going to have his genome sequenced anytime soon? There's absolutely no way he's going to have his genome sequenced anytime soon. I mean, they don't have access to any of this kind of stuff. And the fact of the matter is that all the models that are made to calculate my risk of atrial fibrillation are based on studies that are done in European populations. So of course it's going to work really well for me. But I've just demonstrated that there's an allele out there that has an extremely large phenotypic effect. The allele's at 26% frequency in a population that's distantly related to Europeans. And it's not in any database, and nobody had ever found it before. And it has an effect on a visible human phenotype. Right? And you can imagine the number of disease-causing alleles that are out there that are affecting disease status, that are segregating at intermediate frequency at populations that are underrepresented currently in medical genomic studies, that are not going to be taken into consideration in the calculations of these risks. Right? Which I thought was kind of interesting as I was putting the slide together. I noticed, to ensure that the information on this page is as accurate as possible, please select your ancestry. So I didn't actually, I should have selected my ancestry again. But you see that they are making some kind of a claim that you can set your ancestry and it'll recalculate these things. But I can assure you that if your father is from Malaysia and your mother's from Zambia, that this is going to be a very, very poor predictor, a much worse predictor of your disease status than it is if you're of European origin. And that's going to be the case for a very long time to come, because the basis of all this information is derived from populations of European origin. Yet we've demonstrated that sometimes alleles that have large phenotypic effects that are segregating at intermediate frequency in populations that are in poorer nations that are neglected by genomics research, these can have really, really strong effects. And we expect that probably to be the case for lots of disease-causing alleles as well. And so the age of personalized, and that's not only threatening to increase healthcare disparities in the future of personalized genomics, that threatens to increase healthcare disparities between rich and poor nations, but also between marginalized populations within Canada and the European-derived populations within a place like Canada. When you go to the doctor in 10 years from now and they look up your genome sequence, if you're an Aborigine in Canada, the prediction is going to be way worse than it is if you're a European, right? I'm not suggesting that I have a solution to the problem, but it's definitely worth pointing out that the Eurocentric focus currently of medical genomics research really does threaten to increase healthcare disparities in the future if we're going to be relying on that type of information to make recommendations to patients. So, and it's worth considering, consider our evolutionary history. We are one little tiny population, the Europeans, and there's lots of genome-wide association studies coming out in Chinese now, are doing a lot more in Asians, but it's basically the nations that are able to fund this sort of stuff. We're just a small little group that has recently come out of Africa. Most of the world's diversity is still found within Africa. Right? And so we're just a population who has recently come out of Africa, and we're like a little tiny subpopulation of the entire Earth's diversity. And Africa contains most of the human genetic diversity in the world. It is completely untapped for the purposes of medical genomics. Right? Yet, that's probably the place in the world where, honestly, that suffers the largest health burdens. Healthcare burden. So it's worth taking into consideration this enormous potential of the genotypic and phenotypic diversity that we see around in the world's populations, and consider what kind of predictions are we going to be able to make for these different individuals in the future of personalized genomics. And those predictions are going to be very, very different between populations. And it's not like discovering the blonde hair allele in Melanesians, put the nail in the coffin to this argument or anything, but it was a way of streaming a red line through the entire talk and giving you something entertaining to think about. But I think it is an important point, and that's what I wanted to share with you today. So thank you very much. These are the folks that helped me out with the project. I was really fortunate. I was working with Ed Buckler when I got the grant, and he works on corn, and I was working on grapes. And I went into his office. I said, Ed, do you mind if I take 6 weeks off and go to the Solomon Islands and do a project on blonde hair and Melanesians? And he's just typing furiously at his computer, and he goes, is it good science? And I said, yeah, no, it's good science. He goes, go ahead. And that was it. So I got to go. And so he was really nice about it. He's not even on the paper. So, but I worked a lot on this project with Carlos Bustamante at Stanford, and my friend and colleague Nick Timpson came with us to the Solomon Islands and helped out, and my PhD supervisor really set everything up, Mark Stoneking in Leipzig, Germany. So thank you very much for your time. I'll take any questions.
What are your thoughts, going back to 23, about using ethnicity to shift the genome results? I mean, ethnicity is societal construct. We often use genome-wide results to decide on ethnic origin. Which way should we go, based on which think they are, or which way the SNPs tell them they are, when it comes to assigning relative risk?
Yeah, I think if the genetic data are available, then that's an objective measure. We see this in this. There was a paper out recently about reported, estimated African ancestry among African Americans. So you get an African American, and you ask them, what proportion of your genome do you think is African, and what proportion do you think is European? And ask them that, and then go and actually measure it, collect the genotype data and measure it and plot that out. And it turns out that the correlation is really, really horrible. And basically all they're doing is a lot of people just judge their African component by their skin pigmentation. While skin pigmentation is controlled, there are a couple very large effect alleles. If you happen to get them, then you end up having very dark skin, and if you don't, you don't. But it's not representative of your entire genome. So I think in cases where you do have genome-wide SNP data, then it's a no-brainer. Absolutely, you make the ancestry estimate using but often it's not the case, and they walk in and they say, you can tell that they're African American. In the case, this is what I see in the literature anyway, they're African American. You ask about their ethnicity or their ancestry. But it turns out that self-reported ancestry is often...
Since 23andMe has genome-wide, why are they asking you to decide?
Why are they what?
Why would 23andMe be asking you to identify ethnicity if they have whole genome anyway?
Yeah, you'd think they'd be able to set your, what did it say? Set your something? set your ancestry. Yeah. Maybe it's allowing you to change your ancestry based on what you think. Yeah, that's a good question. I don't know why they need to set it if they knew it already. But yeah, I could show you my ancestry too. It's pretty cool. I'm just European, standard, bland European guy. Super boring. It would be, I think it's more interesting for people who are of admixed origin or are curious about, I think I have one grandparent who's Native American, and I have another grandparent who is African American. I'm kind of curious about what proportion of my genome is each of these things. I think that's a really interesting thing. But.
You did skin pigmentation measurements in your Melanesians. Was there I don't think you showed us the data, but was there any sort of correlation with the
hair?
With the genotype?
Oh, with this genotype?
Yes.
Yeah. So there is, but so in order to really do that properly, to find out whether it is really a causal allele, you'd really need to, we genotype the single SNP, right, in 1,200 individuals, and then we have skin pigmentation for 1,200 individuals. Yes, there is a significant correlation between that particular SNP and skin pigmentation. But we know that skin pigmentation is also super highly correlated with ancestry, and that you're not correcting for population structure in that analysis when you're just looking at a single SNP. So really what you'd want to do is have genome-wide SNP data from the entire cohort, from all 1,200 individuals, have full SNP data. And then you could run the standard mixed model or whatever you use to correct for population structure. Skin pigmentation is infamously correlated with population structure. And so running a single SNP against skin pigmentation doesn't give you a very reliable result. So it's one of these. So we do mention it in the supplementary material and say, yeah, it's correlated with skin pigmentation, but I bet you if you I bet you 90% of the SNPs in the genome are correlated with skin pigmentation, because skin pigmentation is correlated with population structure, and genetics are correlated with population structure. So in order to correct for that, you'd really need to collect genome-wide SNP data, and we don't have that data. We still have 1,200 DNA samples and all that phenotype data, height, weight, circumference, all these different measures from these Solomon Islanders. You could do a genome-wide association study for all those traits, but you know, we work on apples and grapes now, and you know, it's kind of fallen by the wayside. But if anyone has, you know, 1,200 SNP chips lying around and they want to get rid of them, then we could always run them.
You could do a pooling experiment instead.
Yeah, you could do pooling. I think, you know, one of the coolest phenotypes in this cohort, because we got photographs of every individual. We didn't measure their hair curliness, but the population segregates for curly straight hair. And that, I'm not going to put as much money on it as, like, I put a case of beer on the whole blonde hair being Mendelian, and I won it. But I don't think curly hair is Mendelian, but I think it might be a large effect of alleles that you could probably pick up with pretty small numbers.
Why did you do an association study for this? I presume you could have got the same result with like 3 families.
Yeah. You could have just done linkage mapping, right? But we were sort of interested in the population genetics of it all anyway. And when we went to Stanford, and you know what? From our experience of trying to collect family data in the Solomon Islands, it was a massive disaster. Because even just people, like what they call their father and their cousin brother and their brother cousin and their, you know, it got very, very confusing when we started going through trying to actually generate pedigrees last time we were there. And we actually collected Y chromosome and mitochondrial DNA data, and then could go back and say, are they really brothers? Are they really? And we had about 11% of the data was wrong. We were misassigned, you know, where people are supposed to be sharing a mitochondrial DNA sequence, they didn't, or a Y chromosome sequence, they didn't. So we thought, oh, we'll just go and we'll just do a GWAS instead.
Did you do some of your, so the hair color, I guess, would be categorically, category 1 or category 9?
For the genome-wide association study, we did, but if you do look at it, it's rather continuous.
Well, some of your things, you know, skin color or height and circumference and so forth, those would be continuous data.
Yeah.
Did you do your association using continuous data?
No, so the only ones, we chose 45 blonde and 45 dark-haired individuals, and that's the only SNP chip data we have. So
some of those might be fairly, you know, things like cardiovascular disease or factorial, and you have continuous data with hard to analyze.
Yeah.
It's challenging to analyze, but you may have a good model there to try some continuous data analysis on something that may end up only having 3 or 4 different
totally.
might be a nice model.
Yeah, and we aimed, when we went there, we thought, let's try to get 1,000 individuals. That's going to be the absolute minimum you're going to need in order to be able to publish a genome-wide association study for a complex trait. Because we're thinking that all the other ones are complex, there's many genes involved, and you're not going to find a single allele. So we wanted to get 1,000 individuals. We got 1,200 individuals, but we've never had the money to actually genotype them all. So if, you know, or, you know, we could do a pooling sort of thing if people were interested in a particular trait. But we have BMI because we have height and weight. But nowadays, when you look at the, you know, genome-wide association studies in the literature, we have like cohorts of like 140,000 people, and you know, so the effects that you can estimate in the European population are, you know, super tiny. The ones that you'd be able to get with 1,200 in the Solomon Islands would be very, very small. But for the ones that you'd only be able to get large effect alleles. But I don't know, if it's not really my domain anymore, but if people were interested in actually giving it a try, we've got the DNA samples available. The Genome Atlantic might be interested in, you know, supporting this kind of endeavor.
Could have mild albinism, do you think? Seeing as the genes involved in the pathway?
Yeah, it's funny, and I don't really understand the biochemistry of this particular mutation well enough to know why would it affect hair but not skin.
Well, there's sometimes some temperature-sensitive mutations, except it's the opposite, that cause albinism. The Siamese cats, for instance, have a tyrosinase gene mutation that's temperature sensitive,
Right.
such that the extremities develop dark pigment after they're born white, but
Right.
with time they develop pigment on the tips of their ears and their paws and their tails. Or it could be like some of the P gene, some of the albinism caused from
Yep.
P gene mutations in Africans that caused things were not so politically correct, but they were called yellow mutants
Yeah.
in African people of African descent. So it could be something similar.
Yeah.
Under.
And as far as we can tell from the skin pigmentation data, that there is an effect of this particular mutation on skin pigmentation. You can look at the correlation between hair pigmentation and skin pigmentation as well.
Yeah.
There's no obvious effect there. It's not like lighter
So you can
skin.
Because I was looking at the pictures, and of course it depends on the lighting, and so
Mhm.
but I wondered if it correlated with
Yeah.
the hair color. That's a thought. And there are some forms of albinism that the mutation causes very mild vision problems. So, I mean, I'm presuming they didn't have significant vision problems, so it just didn't come up, but it's still possible you might have, actual albinism without having significant vision loss just because it's a mild mutation.
Mhm.
So that's about
Yeah. What other effects this mutation might be having? Very difficult to say, but.
One of my hobby horses again, I'm not sure they've been disadvantaged by not having this technology available to utility in Western culture. They sequenced Dr. Quest from beginning to end. Told him he should be on the only thing that came out of it was that he should be on a statin, which he decided not to take because his protect was not like this. So, one of the first those sequence happened.
Right.
That great study on the Dutch where they looked at height on the GWAS data and compared it to taking differential height. And differential height was much more accurate than 50%.
Yeah. Yeah.
So, I mean,
That just means the models aren't good yet.
and interested in good high speed fit, but we will apparently drink beer.
Yeah. Yeah, no, I agree that the models aren't all that good right now, but we're also not dealing with whole genome sequence. I think from my naive perspective, personalized medicine, I think a lot of what they're going to focus on is those rare large effect mutations that they can detect with whole genome sequencing, not things that segregate at 50% of the population and have an effect size of 0.2, right? I think they're going to be looking through your genome, and even we saw that paper last year where they even fetus of 8 weeks or something. You sequence the entire genome of a fetus of 8 weeks, right? And if you can do that and you find that particular mutation, it looks like de novo arose in the mother. It looks like it's likely to cause Duchenne's muscular dystrophy. Then they should get the same kind of counseling that other individuals do who have it as part of the family history. But that's the slippery slope of so some dude in some lab made some prediction about what that mutation is going to do, even though we've never seen it in the human population before. But it does seem to suggest that it's going to knock out function of some particular protein that is totally critical to the cell cycle. What are we going to do with this fetus? That's where personalized genomics is going to head into all that murky ethical territory. But in terms of making predictions for adults right now on what my risk of type 2 diabetes is, yeah, admittedly really, really horrible predictions.
Well, even simple genome, look at the sequencing of the CNT of alpha, down to beta, and the sequencing of the genome before. They found out he was a carrier ABC of a external genomic dystrophy mutation, which would be completely obscured by a CNT.
Mm-hmm.
And what did you tell his daughter? I have no idea what they told the daughter, but that's not a lot of people. But, grandkids are at risk of a very unpleasant disease if the identification is correct. And that's one of the first things we sequence.
Yeah.
I'm sort of skeptical that personalized medicine, for many important things, for example, arthritis, many other chronic traits where there's multiple genes interacting, stochastic factors. I'm somewhat skeptical that we'll ever be able to predict with anything more significant than a 10 to 80% risk. Those are bare bounds.
Right. Yeah.
So I'm not sure it's a model problem. I think it may be in some cases it certainly will be, but for many important diseases, my sense is, and this is purely intuition, may be wrong, may well be wrong, that we'll never know. I mean, you can take the example, for example, of paired organs, where you get two eyes, you're born with identical genotypes, and yet they develop, say, macular degeneration or some other serious disorder at different rates.
Right.
It's not predictable from a genome sequence of the organism.
Yeah, in the end, it's a matter of heritability, right? Like if it's a heritable trait, we know height is highly heritable, and now we can predict, we account for a certain amount of the variation in height. We should be able to predict height. If we look at your genome, we should be able to predict how tall you're going to get, right? Because height is really heritable. If your parents are tall, you're going to be tall. And you should be able to predict from your genome better how tall you're going to be than just by measuring the height of your parents. But it turns out when you measure the height of your parents, you're also picking up on the environmental factors that made your parents tall or short. So in the end, your type 2 diabetes, prediction about type 2 diabetes is going to probably be better if you just clocked in how many potato chips they eat a week is going to be a better predictor than their whole genome sequence, I suspect. But yeah, it's because the heritability of a lot of the traits that we're interested in terms of chronic diseases in first world nations are really low. And we're just picking at these like tiny little effect sizes in nature genetics now that are ridiculous. And I think there's definitely utility to it, and at least people think there is. But in terms of making whether the model's going to get really, really good and we're going to be able to predict whether somebody gets type 2 diabetes, well, you can't predict it if 95% of the variation is environmental anyway. So.
Well, that sort of makes your other title sort of ironic, because if you look at the non-Caucasian populations as being disadvantaged because they're not represented, or actually they might be advantaged because they're not going to have to spend increasingly more money
On healthcare, getting bad predictions about their health. Yeah. Yeah, no, it is possible. And I'm not the final arbiter on whether personalized genomics is going to be a go or not, and how well it's actually going to work. I think the people who are investing billions of dollars down in California into the whole endeavor are probably going to be the ones who decide. But the other thing is they'll probably end up making money even if their predictions are bad, right? Because they'll figure out a way of doing it. But,
yeah, I think it may influence interpretation of data, so it may become sort of something that it's used to determine, how people will be covered for their health care.
for the health insurance things.
Exactly, even though the predictions are not very good.
Mm-hmm. Yeah, it's a really problematic territory because the commercial side has really picked up on it and gone as far as they can push, while the academics are sitting back and going, "Wow, those are like, where did that number 46% something atrial fibrillation? Who came up with that number? And why am I looking at it going, and I'm a genome scientist, and I'm looking at it going, wow, Jesus, at the top of the list, and it's a big number." Right? Right? So I imagine everybody else who knows nothing about genomics are going to look at it and go, whoa, I got a really high risk of atrial fibrillation. Gee, I better Google that. Yeah. So, yeah, but yeah.
I wonder if you could speculate on why the population frequency is as high as it is for that. Is it that these individuals are looked at as a very attractive in terms of being a partner, or does he think it correlates with something that's protective in terms of survival?
Yeah.
What do you think?
So the genetic evidence, we don't have any evidence that it was selected for, that there was any advantage at all to having blonde hair. But the way of testing that is very difficult because it's a recessive allele. So even if it was really under strong selection, we might not be able to detect it either. The other thing that you can do is you can go around and you can ask people, and you can say, hey, do you find blonde haired people attractive? And we did that, and we asked a lot of people whether they preferred people with blonde hair, and the answer was sort of, some people were like, yeah, I like blonde haired people. And I don't know, not really, and not any more than anybody else. So there was definitely no definitive across the board preference for blondism. And then other than that, to evaluate that hypothesis is very tricky. So I don't really have a strong personal, I think it's completely possible that an allele rises to a frequency of 26% by drift. That it affects such an incredibly visible human phenotype and wouldn't either be selected against or selected for. You imagine that first blonde kid who came out. Imagine we're human, right? You either kill them or you have sex with them, really. Right? You're either going to desire them or you're going to try to get rid of them. You would think with such a striking visual feature, but apparently not. At least there's no evidence that it was one way or the other.
It's something like what happens with Pimela Islands happened here, for instance, if there was a big tsunami at some point, very few survivors, and
Yep. So
something like that.
there is a bit of a founder effect in the population in general. They do have higher IBD, like identity by descent across the genome, than you would expect in outbred populations. So, we don't like to call them inbred populations, but they do have, more stretches of the genome in common with each other, long stretches of the genome in common with each other than Europeans do, for instance, in a normal European cohort that you would use for this kind of study. It's one of the reasons why our region was really quite large, the IBD region. So, so that does contribute to the rise in frequency to 26%. But we don't have like a full, accurate demographic model of how this population evolved. And you don't know the
historical event that
Right.
suggested decimation of the population.
no, none. There's nothing like that. Like not really
before,
recent.
Before history.
Yeah,
History.
sure, yeah. But they, some of their ancestry has been hanging around the Solomon Islands for 30,000 years. So,
Predate.
yeah, it's not like Iceland where you have a very recent migration and population bottlenecks.
So was there any variability in eye color?
Yeah, so there is a little bit, but I think, and I don't know anything about eye diseases and things, but I think some people have that kind of cloudy blue eyes from certain eye diseases and stuff. So we were starting to look into eye color, but what we noticed is that it was more frequent for this sort of cloudy blue eye thing out on the outlying islands where they don't have, they have some of the nutrition is a little worse, and the access to medicine is worse as well. And people were saying, that's no, they have brown eyes, they just have something wrong with their eyes. But we didn't, after a while we were phenotyping, we were like, let's forget the eye color thing, man. Like half the people, how do you score eye color? It was difficult to score, and we were just like, let's leave that one. But, and 95% of the people that we saw, everyone has brown eyes. Yeah. So, they're very dark eyes. Eye color is kind of very tricky to score too, because it really is, it's quite quantitative, at least in Europeans, and you see every possible shade. So. Anyway, I think I've spoken enough.
I think that time is up, but thank you very much, Sean, and for
Thanks.
someone who has a personal interest in human diversity and hair color, haha. Thanks very much.
A rare meeting.
We really appreciate it. Not that rare, right? Nova Scotia.
Less than
especially.
26% frequency.
We love us in Nova Scotia.
Yeah.
Okay, so please just fill out your evaluation form if you want to get CME accreditation, and also we like anyone's evaluations, please, no matter if you're going to get accredited or not. And next talk is with Jason Berman and zebrafish, use in genetic disease research. See you then.